Good morning. So, in this part, we are going to discuss about the enzyme linked receptor. Now, in case of the enzyme, the kinase linked receptor, this is the transmembrane receptor because it is present. You can say that in the membrane, and you can this uses the second messenger signaling that triggered the cascade of the cellular event. Now, for this particular type of the receptor, they contains large transmembrane protein. They contains the large transmembrane protein which spun with the width of the cell spanning consisting of the alpha helical coil. So, alpha helical coil means this particular area, this alpha helix that will discuss later. So, they contains of the large transmembrane protein uh, which span the width of the cell membrane consisting an alpha helix coil. Extracellular domain of this particular membrane consists of the protein binding site. It is near to the N terminal where the ligands which will bind in order to relay the signal. Here it will relay the transmitting the signal. Next is intracellular domain has a abundance of the tyrosine residue. Generally in that majorly tyrosine residue it will be present that phosphorylate each other to become active and serve as a docking platform from the different cytoplasmic protein that involve into the signal transduction. So mainly you can say that phosphorylation of the tyrosine molecule it will happen after binding with the protein ligand, and then different proteins are there which is involved into the signal transduction. Now, generally, these receptors shares a common architecture, the structure. Of course, you can say that through the different mechanism, and large intracellular ligand binding domain are connected via single alpha helix to the intracellular domain. So, there is a intracellular part and extracellular part, and this extracellular and intracellular part it will be joined by the alpha helix. These are the receptor involved into the event which are controlling the cell growth, cell differentiation, or you can say that they are indirectly they are also regulating the genes transcription. Now, for example, this if we are talking about the this is the you can say that that particular receptor, we can find that here we can find that there is a extracellular domain and intracellular domain. This is the tyrosine molecule. So phosphorylation of the tyrosine molecule it will takes place inside the cell, and then mainly here we are showing that three different mechanisms through which they will target the gene. Now what are the you can say that signal pathway: MAP kinase signaling pathway, JAK-STAT signaling pathway, and that is the PI3K-ATK1 signaling pathway. And through this pathway, you will cause the gene target, and then it will cause. You can say that we have told that they will cause the. Here we have already told they have indirectly regulating the genes transcription, and also we have told that they require the they uses the second messenger signaling, the second messenger signaling pathway. And there is a different types of the receptor, or you can say the signaling pathway is there, like receptor tyrosine kinase, then receptor serine and threonine kinase, receptor guanyl cyclase, and tyrosine kinase associated receptor, and receptor tyrosine phosphatase. So these are the different pathways are there. Now we'll discuss about the some of the, not all, some of the pathway. Like first we'll discuss about the growth factor. Now growth factor it will bind to the growth factor receptor which is the enzyme linked receptor. Now what will happen? See, this is the intracellular domain, this is the extracellular domain. In the extracellular domain, the endogenous substance or ligand it will bind. Now these two part it will be linked with the transmembrane helix. Clear? Transmembrane. This is the tyrosine kinase domain, and this is the tyrosine residue. Whenever the growth factor it will come and bind, here you can say that conformation changes or the dimerization. Clear? Dimerization. and after the dimerization, if you see that there is a autophosphorylation. If you consider here autophosphorylation, and with that the binding of the SH2 domain protein, that is GRB2. This is also a protein, so GRB2 it will be binding. Clear? When there will be the GRB2 will be binding, again the activation of the GTP and GTP exchange it will take place. So RAS it will be activated, and then GTP to GTP activation, GTP to RAF, and again phosphorylation MAC and MAP kinase. Already we have told about the MAP kinase, and there will be again the phosphorylation, and this phosphorylation trigger the genes transcription. Whereas if we see about the Jack and Stat pathway, the cytokinin it will bind. Already here we are showing the conformation change and the dimerization it will be there. There is no tyrosine kinase molecule, it is there. Long tyrosine kinase domain is there, but tyrosine residue is there. But then what will happen? The conformational changes for the binding of the Jack. The Jack it will come and bite. All these are the proteins. Jack it will come and bite. Jesus kinase proteins it will come and bind. Then there will be the phosphorylation of again tyrosyl molecule. After the phosphorylation state, it will bind to that phosphorylated molecule, and that will trigger the genes transcription. So this is again one type, you can say that one of the mechanisms. So there are different growth factor, insulin, cytokinin, these are the different endogenous substance which acts through the tyrosine kinase, tyrosine residue phosphorylation. That means these are the kinase link receptor.